For those of you, most of you know, I think, we're a public-private partnership. We were formed about 4 years ago. We're funded by the ag industry, and we're a university entity. We're tasked with really bringing resources and bringing focus to the problems of industry, and we do that largely working through U of A researchers, but as you'll see today, other researchers and other resources too. So certainly one of the top priorities they gave us to work on was Fusarium Wilt, and we've been doing that. And maybe I'll just recap some of the efforts we've done over the last 4 years to set the stage for where we're at. We brought in about over $300,000 of research dollars to fight it. We started with an international symposium back in 2015 here in Yuma, which brought together growers and researchers and a field trial. On our website, we still have all the PowerPoints and the videos and everything from those conferences. So if you want to see what came out of the conferences, the planning documents, the brainstorming, the video presentations, they're all on our website. So it's a good resource. Rosa Bevington's over here. She does a great job putting that all together. We've done field trials 4 years in a row working with Dr. Matheron and Dr. Pryor. So that's been looking at resistant varieties, looking at chemical and biological products, looking at cultural practices, things like that. Those results are all on our website. Unfortunately, not a lot of them talk about things that work, but they at least tell you things that don't work. And there is some progress being made, hopefully, on things that work. What else? We've worked with trace genomics to look at trying and other ways to try to figure out if you can sample the soil and figure out the disease pressure that would be expected in a field before you plant, so that you can make planting decisions based on that. And there's been quite a few other efforts. And so this year, in 2019, we're kind of supercharging our efforts. Number one, because growers are saying it's becoming more and more of a problem, unfortunately. And number two, we brought on Dr. Stephanie Slinski as our research director. And so, yeah, this year I'm really excited with where we're heading. Cooperative Extension, you'll hear in a minute, has also brought on a plant pathologist, Dr. Bindu. And so, yeah, there's going to be a lot of effort on it. And Stephanie, just a quick way of introduction for Stephanie. She came here from Florida. She was at a somewhat similar organization working on citrus greening disease back there. But she has her master's in microbiology and a PhD in plant pathology. So this is kind of in her wheelhouse, and she's been putting together some strategies for moving forward on this. She's going to fill you in on projects that she's got planning, and her efforts and her collaborations have led to a lot of the speakers that are here today. So I guess with no further ado, I'll introduce Stephanie to talk about some of our plans and introduce the speakers and everything for the rest of the day.
Thanks everyone for coming. I'm really excited to hear these speakers this afternoon. As Paul said, Fusarium Wilt's a high priority for us, and I know it's a high priority for you to find some solutions. And in this coming year, we have several projects that are in development. The trials to screen varieties and chemicals that Mike has been conducting continuously since 2015. We're continuing these trials. We're working to get some new materials and new varieties in these trials, as well as continuing some of the varieties and chemicals from previous years to get more information on them. I'm also developing a project looking at some cultural methods to decrease the population of the Fusarium in the soil. Thank you. There's been some success in Yuma with solarizing soil, and this technique called anaerobic soil disinfestation, or also biosolarization. These are techniques that can ramp up that the spore killing ability of that technique. It's been used in other parts of the U.S. Steve Koike, one of our speakers, has been involved in some of this work, and I hope to talk to him more about it in the future. It's also been used in Florida, in the Netherlands, and in Japan, and even in commercial situations. It's similar to solarization, but it's with an added carbon source, and then it also has a low oxygen environment, and it suppresses soil-borne fungi, nematodes, and weeds. The limitation in these methods in other areas is time and temperature, and in Yuma, there's the summer where you're not planting any crops, and there's also the high temperatures. So it's a really good possibility for the Yuma area. And we hope to get a pilot project in the ground this summer and a full-scale project by next summer. Something else we're working on is for all of the projects that we want to do, we need a reliable quantification method for Fusarium in the soil. We're assisting in the development of new tools. We're working with Dr. Frank Martin, who's the speaker today, to help him develop molecular tools. Dr. Barry Pryor is helping to develop some tools. So we're trying to assist these efforts and bring these tools forward to you. These are important for research, but they're also important for you, for disease management. It can help us develop thresholds so you know what crops, what level of resistance you need to have to successfully produce a lettuce crop. And to do this, we do need to develop some thresholds, so they're usable tools for you. And from all this, I'm talking about developing projects. You might think I'm a researcher, but I'm not. That's one of the limitations that I have. I can't go out and do all this research. So I'm spending a lot of time trying to find researchers to conduct these experiments and field trials. Mike Matheron, unfortunately, is retiring at the end of this calendar year, and we're working with Alex Hu to hopefully develop some projects. He came to the University of Arizona in 2017, and he's interested in Fusarium Wilt. We hope to work with him in the future. Barry Pryor, he's been working with Fusarium Wilt for quite a few years now, and we hope to work with him more. And the extension just hired a plant pathologist. We're very excited that she's here. She will be running a diagnostic lab and maybe collaborating on some of these projects. And I've also been talking to quite a few people in California about different methods and maybe new collaborations. So what I'd like to do now is have Russ Engel come up and introduce Dr. Pudell.
All right, well, thanks. My name is Russ Engel. I'm the director of Yuma County Cooperative Extension, and I just wanted to take just a short, real quick here. I wanted to introduce the newest staff member that we have. This is Dr. Bindu Pudell. We hired her. She's a plant pathologist. We hired her to increase our capability to deal with and handle the issues associated with plant diseases. So I guess all I wanted to do is like I said, introduce you to her, and would you all please, introduce yourselves when you get a chance, talk to her. If you have any plant disease problems or issues and you'd like her to come out and look at those, please contact her, get a hold of her. She's currently trying to kind of set up and improve on our lab, so she's just trying to increase our capabilities of dealing with plant diseases. So, like I said, if you have any need for her, if you have any questions, problems, issues, give her a call, and she'd be more than willing to come out and work with you and try to help you with some of those issues.
Steve Koike is the director of TriCal Diagnostics in Hollister. He and his staff operate a plant disease diagnostic lab. They test soil for pathogens, conduct research on soil-borne and foliar pathogens, and provide plant pathology and crop production information to clientele who grow and work with various agricultural and horticultural commodities throughout and beyond California. Previously, Steve was the Central Coast Extension plant pathologist, which is when I first met him, with the University of California Cooperative Extension, where he operated a diagnostic lab and conducted a research and education program on diseases of fruit, vegetables, and ornamental crops. Thank you for coming, Steve.
Thank you, Stephanie. Well, it's a pleasure to be here. Thank you for inviting me, and I'm glad to have a chance to tell you a little bit about what's happening in California and review a few things. First, I want to tell you, when I came into this room, I was really impressed because I saw this is really a top flight organization because they have picture postcard of diseases. I thought this is really a cool idea. And then in the back, I just saw it's an advertisement. So, but Stephanie, I might steal this idea. Picture postcards for plant pathologists. Don't you? That'd be a big revenue generator, don't you think? Okay. So Stephanie asked me to talk to you this afternoon about some of the things happening in California. What I'll do, I'll briefly cover some of the basics on Fusarium wilt of lettuce. And I know a lot of you, and a lot of you are super experienced, so I'm not pretending this is new information, right? We're all together on this. Most of you know what's happening and you know the details, but this will be a way to pull it all together. And maybe there's a few nuances here and there you're not familiar with. There's some research we've been doing that we'll add to the mix. And then don't see too many people strictly growing in California, so I can update you on what's happening in California, which, if you don't know already, is not good news for us. So with that, let's see, slides here, right? Okay, so again, a quick review, some basics on the disease and the situation in California, and then I will emphasize some things about diagnostics, which again, you guys are very experienced. Mike, you guys know, but I just want to touch on that because it's very important. And I'll preface it by saying we have some really outstanding experienced growers and PCAs in California, as you do here, and there are misdiagnoses happening with this disease. So I think it's worthwhile just to review that. So this is what we want to see, but we don't see it all the time. So quick history on the disease. It's not a brand new disease. It was in Japan first, as far as we know from reported literature or records. Then the first time outside Japan, interestingly, was us. So we have that distinction of being the first place for lettuce fusarium outside Japan. Then 2001, you had it here. This is Mike's work. Mike was the first one to document that in Arizona. I may have the year wrong, and maybe the year before, maybe 2015, I'm not sure, but it's in Florida. I flew to Florida last year and talked to their group, and they've been following what you and we are doing out in the West. They're very concerned about lettuce in Florida because they have Fusarium wilt on lettuce in Florida now. And in most significant lettuce growing areas, we have this problem. So let me tell you what's happened in California. Again, some of you know this already, but again, 1990 was the first hit outside of Japan. That's the Huron area in Fresno County. It's a small in-between growing period between our big Salinas industry in the spring and summer, and then the desert area, which includes you for the winter lettuce. Huron kind of fits in between there, and they had it for the first time in 1990. Then we had it in the coast, 2002. Our group was the first to find it in Salinas Valley in 2002. And then the picture gets, unfortunately, more crowded here because we're very concerned about the future. So 2005, you can see it's starting to spread on the coast, and then I'll bring you up to date now. That's what the map looks like in coastal California. It's kind of all over. I'll have another map of just Salinas Valley I'll show you in a minute. But this is why we're quite concerned, and the chance to talk to you about it and interact with you, Stephanie, and others, it's something that is very important to us as well. I'll tell you a quick story. Long time ago, I floated this proposal to work on Fusarium wilt with the lettuce board in California, and that was 2005 or so. And a couple of the big growers in Salinas said, Steve, we like your work, we'll fund you, but that's really a desert problem. We don't have it here. You can, we'll give you a little bit of money to work on it, but it's not really a desert problem. Well, you see that map there? It's not a desert problem anymore, strictly speaking, and those growers now were giving me bigger money because it's a coastal issue too. And I would say at the time, we sort of thought the same thing. We thought, given our coastal climate, so cool, our lack of history about it, it's not going to be a big deal. We are going to be worried about verticillium, which I'll show you. That was our big thing. It's not Fusarium, it's verticillium. Well, now we have both, and it's a big deal. Some real basics here. I'll be quick because of time, but you know this already. So it's a soil fungus. It is host specific to lettuce. It only goes to lettuce. It can be seed-borne, and that's Barry's work and others, but Barry work on the seed. Although on commercial seed, it's quite rare. I don't know if that's changed, Barry, but I think it's very rare, yeah. So it's not a significant source of inoculum. And then it does survive in the soil as a clematospore. Those are the structures up at the upper top there. That's why it sticks around, because it survives in the soil as these structures. Although, interestingly, it doesn't stay around forever. I'll show you the next slide, this graph. But Tom Gordon did some nice work. after almost 3 years, you get really a big drop on inoculum. So if you're not going to plant lettuce anymore, which we in Salinas, of course, we never do that. We always come back with lettuce. But if you do stay out of lettuce for a while, the fungus does drop pretty quickly. Whereas verticillium, that's like an 8-year. For verticillium on lettuce, you need to not plant lettuce for 8 years. So it's a problem because we grow a lot of lettuce and grow too much lettuce. So I don't think any of us are going to stay out for 3 years and plant no lettuce, but it shows you an idea here. There's some possibilities in terms of rotations. And then for you who are breeders or for you who are buying varieties from breeders, the race picture is very important. Right now in California and the West Coast, including you, as far as we know, only race 1. But there's some diversity there. Japan has race 2 and 3. There's a new race 4 in the Netherlands or Belgium, one of those places reported 2 years ago, I think. So some diversity, and that will complicate things for breeding, and especially if these races get around, that's further trouble for you and me. Here's that graph. This is that graph about the 2.5-year survival period. This fungus doesn't last forever in soils if you don't plant the host. So you can see there's a significant drop based on Tom's work here, so that's kind of interesting news. Although there's a footnote to this too, and I'll show you that in a minute, because it's not that simple, which again, I think we all realize plant disease issues are rarely really simple. So what does it look like in patterns? I think this is probably familiar to most of you. Being soil-borne, it pretty much is being spread as introduction to fields on equipment. So mud, dirt, dirty equipment, crews, they will come into fields and they'll drop contaminated mud or dirt in a certain area. And that's where you first see the problem, right? It's not something that hits the whole field. It's going to be in a corner or this strip here or this patch here. See that top photo? There's maybe 2 or 3 patches. Very limited area, but that's where the contamination took place. And then over time, as you keep planting more lettuce and you move the ground, then that's what's going to happen is this sort of distribution where it moves out from that center. Typical traditional soil-borne issue starts with an introduction in a corner somewhere, and you move that with your tractors and dirt and stuff, and pretty soon your whole field, or at least half the field, is going to have this problem. I'll be quick again on symptoms. You probably are very familiar with this, but just to cover it. So basically you're talking about a vascular disease that stunts the plant, so a healthy plant on the side here on the left, and then stunted plants on the other side. I did this photo on purpose to emphasize to you this is not a root rot disease. And this is very important. Again, we have some great professionals in the coastal area. They've made some misdiagnoses with a root rot disease in Salinas, so don't make the same mistake, and I'll cover this for you. But there's definitely healthy roots. Fusarium wilt does not cause a root rot in the sense of a root rot disease, and I'll show you in a minute what a contrast to that. Vascular disease stunting, not a root disease. Then, of course, cut it down in the middle, take a look at the vascular. There's the typical vascular discoloration of a Fusarium wilt disease. It typically is a brick red color, although I want to point this photo here to you. If you were in Salinas Valley and you saw this, you would start to think about verticillium, because verticillium causes a black discoloration in the root. But again, you know how symptoms are, right? They overlap with each other, so you got to be careful. I'm a proponent of testing everything. When I was with UC, we tested everything. Now I'm running a commercial lab, we test everything. A brick red root doesn't tell me anything, we test it, because you can get this ambiguity with color, so be careful with that. But in general, Fusarium is brick red, orange red in the center there. Verticillium is going to be black in general. Then the other thing about infection on Fusarium, which is I think quite unusual, or at least it's novel to me, and that is stage of the lettuce when infected and when it's symptomatic. You can have plants that are starting to show symptoms really early, pre-thin, maybe a little bit pre-thin, but definitely by post-thin rosette. You can have plants that are going to show symptoms and they never develop past the rosette stage because you have Fusarium wilt infecting those plants early. A little bit unusual, I think, for a wilt disease, but in lettuce you have that. Then you have some plants, like at the top there, they're infected late. They're going to get size, they're going to get pretty big. They won't form a marketable head because they're diseased and not forming the head properly, and they're infected, but they got to that stage. So if you see a field that has small plants that are infected and big plants that are showing the signs of infection, and in between plants showing infection, That's another hallmark of Fusarium, whereas verticillium, which again you don't have, we have that. It is a late season. You only see verticillium symptoms 10 days out, 8 days before harvest, something like that. It's very late. Fusarium, you have varying size and really uneven fields because you have some plants infected quite early and some infected late. Okay, so let me mention something about symptoms, okay? Again, I think you're very experienced. I'm not questioning that at all, but we've had great, very experienced PCAs misdiagnose Fusarium. Why? For the following reasons. Some of the really hallmark general above ground symptoms are shared by a number of pathogens, okay? And some of you have seen this slide before. So if you have stunted plants, plants with wilted leaves, plants with yellow leaves, plants that are collapsing, those definitely are symptoms that could be Fusarium. But they could also be Pythium wilt, Sclerotinia, Botrytis crown rot, also verticillium. Those general symptoms fit all those diseases. So if you're not careful, you can confuse what you're looking at if you haven't done a thorough job of your diagnostics. Now, you know, I'm limited time here, so if we had time, we can run through this. There are nuances here, right? We can go through disease by disease, and I can point out to you how you can separate Pythium from Fusarium, and I can point out to you how we can separate verticillium from Fusarium. We could do that, but in general, my point to you is that be careful and don't make assumptions, and my recommendation is, you know, work with a diagnostician, work with us, work with Mike or whomever, but if you're not sure, test it. It's really important to test it, because one of these days, there'll be something new there, and I'll show you some new things in Salinas that have confused our diagnostic picture. So just some things that overlap in general. Again, you know Sclerotinia, right? Very few of you are going to confuse it, but just my point is there are overlaps in symptoms. That's my point. So Sclerotinia is one of those. Verticillium for us, very much misdiagnosed because growers know about the dangerous verticillium on lettuce in our coastal area. They're not thinking about Fusarium, and they'll say, hey, Steve, you know, I've got that verticillium again. I said, well, you know, we should test it for you just to make sure. They'll send it in. It was not verticillium. It was Fusarium. So that happens. So be careful. Verticillium overlap with symptoms with Fusarium. This is the new one, new in the sense that it's a new issue. This actually was first described in the mid-1990s in Coachella, Pythium wilt on lettuce. It's a root rot disease, okay? So this is that one I was talking about. This is one where growers drove by, and no names, in case you're affiliated with those companies, no names. They drove by and they said, gosh, that's that new Fusarium that Steve was talking about at his meeting. I've got Fusarium wilt, and I didn't know I had it at this ranch, but gosh, I got that now, so I'm gonna call Steve, and you know, what am I gonna do about it? Well, we worked with that grower, and it was not Fusarium, it was this Pythium wilt, okay? But again, I think, hope you agree with me, from your windshield, which you never do that, right? No one does windshield diagnostics right here, no one. It could look like Fusarium wilt, very much it could. And the clue here is to dig it up and make sure you get all the root, because this root breaks off near the top and you don't get that. If you dig up the whole root, it is a root rot disease, okay? It's a true root rot disease. And Fusarium wilt, is that a root rot disease, Mike?
What?
He wasn't listening. No, I'm teasing. So Fusarium wilt doesn't rot the roots, right? So, yeah, so you got to separate it that way, okay? And then another one, I think it's down here, I don't know how important it is, but lettuce dieback disease. It's kind of a big time issue in some ranches for us in the coast, but lettuce dieback, the virus disease, tomato bushy stunt virus. Again, I would propose to you the symptoms overlap enough to where you should have it checked out, just in case. And interestingly, some of the varieties with bushy stunt, when they get older, the center core darkens a little bit. See, so there's a little bit of ambiguity there. It doesn't turn brown. I don't want to overstate it. It doesn't turn brown, but it's dark in there, and you say, that doesn't look normal. Could this be Fusarium wilt? Well, see, have it tested, check out, make sure you're doing the right thing with that. And then the last one, black root rot. I don't know, again, if you have this on lettuce here, I think you've got the fungus down here for sure. It goes to cotton, beans, lots of things. But black root rot, it's a root nibbler. It causes a decay of the small roots. The lettuce plants never die, but they are stunted. And then as stress comes, the bottom leaves turn yellow. Well, what does that sound like? That sounds like Fusarium. And we've had misdiagnoses of Fusarium with this disease because of the overall symptoms, okay? So that's my point. I hope I didn't overdo that with you, but I would just be careful. The first point in solving a problem is identifying the exact cause, so I'm big on that. That's what I did for almost 30 years with UC and now as a commercial company. Diagnostics is so important, and I just preach that to everybody. I think it's so critical to make sure we've got that right, okay? Now, having finished thinking about the overlap, of course, the vascular thing does set aside Fusarium and Verticillium, okay? So I tricked you there. I didn't show you those arrows at the beginning, but you could say, well, Steve, the vascular things eliminates the others. Pretty much, okay? So yes, the vascular symptom is really key for those two diseases, and for you, fortunately, you only have the Fusarium, you don't have the Verticillium. So you'll say, well, can't I just see if it's discolored in there and then assume it's Fusarium, right? Would you do that, Mike? No, Mike's not going to do that. He's a professional. So, and see, I'll show you why. There are other things that caused center core of lettuce to turn dark, see? So without knowing anything about these photos, I can propose to you that at least one of these is Fusarium wilt, right? Because of that profile. And you would not be able to tell without testing. Again, if we have more time, I'd like to do a quiz with my PCAs, but we're not going to do that. Top here, that's systemic downy mildew. Recognize this one? You should note the size of the plant. That's ammonium toxicity. I don't know how common it is down here. Ammonium toxicity on lettuce, because lettuce roots are actually sensitive to ammonium building up in the soil. And then these three are physiological disorders. Weird things, lettuce does weird things when there's temperature changes and stress and other things and the variety. The center core will turn dark. It'll kind of look like Fusarium. So even that feature, you should be careful of making assumptions. So I don't know about you, but for me, I just don't like being wrong. So that's my motivation. So that's purely it. I just don't want to look bad. You feel it, right? Right? You're an extension guy, right? Yes. You don't want to look bad, do you? Right. Okay, so have it tested, right? Yes. Okay, excellent. He's good.
Have her tested.
All right, have her tested. All right. We want to drill in a little further. I want to show you again what's happening specific in Salinas. So this is what happened. I think this is about 15 years ago. I don't have the exact date. I should have put that in here. 15 years ago, we had our outbreak of Fusarium on lettuce, and it was strictly a South County thing. And South County is King City, San Bernardino, if you know our area. It's warmer down there, drier, sort of desert-like for us. And so that made sense because we felt Fusarium is going to follow on lettuce. It's going to follow the regular pattern where it's a warm season fungus. It likes warm soil temperatures. It's going to do better and be more aggressive where it's warm. So of course, if we have it at all, it's going to be in our South County lettuce, King City, Soledad South, okay? So fine. Well, here's the bad news. This is today's map, and we were wrong as researchers because we're rarely wrong, Mike, right? Okay, yeah. Well, we were really wrong because we have really severe outbreaks of Fusarium wilt on lettuce in the north part of our county. And if you know our north part of the county, man, you know it's like 58 degrees in the middle of the day, right? And so we have some severe Fusarium wilt on lettuce in the north. So this is now why California, our coastal industry, is woken up to this. We are woke, and so we're concerned about Fusarium as well as these other things. You know, Verticillium used to be the only focus in terms of these vascular things. No longer. We are really worried about Fusarium, which is one reason why I'm here, because we have to learn together and help each other get out of this pickle, right? And we'll talk about why it's in the north. I think we all kind of know why, but we'll talk about that when I finish up with management. The final thing about basics, you know, why this map here, why does this map take place? It's because of the nature of the fungus. So you already know this. These are 5 important soil-borne pathogens, and they all make small, microscopic, tiny resting structures that can survive in the soil for a long time without a host and without moisture. They make these survival structures. That's why they're soil-borne, right? And so here's Verticillium. This is that Pythium wilt on lettuce. Fusarium at the top. Macrophomina, I've circled one microsclerotia of Macrophomina. This is my penny, and this is a grain of sand there. And this is Thalaviopsis, the black root rot fungus. So this is why it's a problem. We spread these things, and it's impossible not to spread it. We have to try and manage that to reduce that, but this is why we have a map like I just showed you. It gets around. The way we farm, we're too intense, we have too much traffic. it's going to move. So that's one development. So I forgot to preface that. This is one development that's new. It's our increasing distribution countywide in the coast, in the Slinski Valley. That's the first development. And then the second new development, we are seeing it on all sorts of lettuce. It used to be only iceberg lettuce, head lettuce, but now we're seeing fields with romaine going down. I had these extension slides I used to talk to my growers a while ago, and under management for Fusarium, I say, well, just don't plant head lettuce because romaine is resistant. You can plant these other kinds of lettuce and get away with it. Well, see, I have to take that slide out. Yeah, I take that slide out because we have romaine going down with it in Slinski Valley.
And the same varieties that it was in the past that it's now taken down, or is it newer varieties that are now going down?
That's an excellent question. His comment or question is, how about the varieties? Has that changed and can account for this? And actually, I haven't done my due diligence on that. I don't know. We would have to go to the records and see, has there been a big swing in the varieties of romaine or whatever? And does that account for this, what I call a new development? So that's a really good point there. But that aside, we used to think just plant leaf lettuce, plant romaine, and we're okay. Just don't plant iceberg. Well, we can no longer say that because I showed you some photos already. We have romaine that's melting down from Fusarium, and I believe that happens down here too. All right. Okay, am I okay on time here? Okay, all right. So, as I head towards home here on my talk, just a couple factors to bring us again all together on the same page. You already know this from your researchers, and if you've seen our stuff, you already know this. I would say there are three significant factors for this disease. So one is the lettuce type and cultivar, of course. Now, we said that romaine is collapsing. That's true. It is still less susceptible than iceberg in general. So it is a type that holds up better versus iceberg. So that's a factor. But still, when you talk about the disease and when we formulate research plans and when you advise each other what to do, you have to consider the variety and type of lettuce. Then secondly, as we all know, as proven by Tom Gordon's work and a lot of other people's work, the amount of fungus in the soil is critical. If you have a high population, you're going to have a lot of disease pressure and issues. And if you have low inoculum, you might be able to keep farming. You might be able to plant some varieties, whereas with the high one, you can't plant the variety. So see this interaction there? Some varieties with low inoculum, you can get away with growing those. But with high inoculum, you won't. So how does that work for you? Well, you have to work that out yourself through your records, working with your breeders, your seed companies. How does the amount of fungus in the soil interact with the varieties? That's an important dynamic. And then on top of that dynamic, you add temperature, the weather, the environment. We all know, again, that this fungus tends to like warmer temperatures. It's more aggressive when it's warm. It does more damage when it's warm. So that's a factor. If it's cooler, you still get disease, still get infection, but it's less economically damaging. So you factor that in. And see, we worked this in Salinas because that one field I showed you that was 50% Fusarium Wilt, that grower came back with the same variety in the early spring and got a great pack out. Got his 1,000 cartons of acre, whatever you guys are trying to do here. Fine. But that photo was taken in September, see? And in September, air temperatures are in the 80s. Which for us is damn hot for Salinas, right? Yeah. In fact, I stopped doing this. I didn't want Mike to get mad at me, but once in a while I check the weather, and when it's 120 down here, I'd call Mike. And I'd say, Mike, it's kind of warm in Salinas today. It's about 72 or something. So, yeah, I took off my jacket today. Right? Do you remember that?
I do.
Yeah. Sorry, I yank your chain there. Anyway, so, but the temperature, so that interacts, it influences your production decisions, because you can plant a certain variety at a different slot based on temperature, or make a different decision on a variety based on high-low inoculum, and how do those three interact? So that's what we're trying to get at here, okay? Okay, and then, yeah, so management, the last part here, specifically about disease management. There's very little new here, and you are very experienced. You could give this part of the talk, and you can outline this. Basically, there's not a whole lot to add, and that's why this is kind of a scary disease. So, of course, sanitation. You got to prevent it as much as possible from being introduced into clean fields. And you got to reduce as much as possible spread even between dirty fields because of the inoculum level, right? Some have a low level. Why would you want to bring in more? See, that's the idea, is as much as possible, try and prevent introduction of the fungus, and that's primarily through infested mud on roots, on equipment, on tires. The seeds in parentheses, that's not an important source as far as we believe. Sanitation, keep it out. Now for us on the coast, we have something called the strawberry crop, which we fumigate. So that is a nice IPM step for us if you can rotate with a crop that does fumigation. So conventional strawberry, if you're going to fumigate that, you do reduce this lettuce problem. And that's why when you are a lettuce grower in Salinas with Fusarium Wilt, or Verticillium Wilt too, one of your best buddies is a strawberry grower, because you ink those agreements and you rotate and swap ground. The strawberry grower is going to fumigate, keep it for 2 years, and give it back to you. And you come back with your lettuce, and the ground has been cleaned up, with parentheses of that issue. So fumigation does work if you have that luxury, okay. Solarization, Stephanie mentioned that. It's an option. It's not super effective all by itself, but it's part of the integrated thing. You have options here, which we don't in Salinas. Of course, the tried and true option of site selection and timing. If you have the option, you don't plant lettuce in the hot field, right? If you have the option. Sometimes you don't have the option. And when do you plant? I mentioned that timing where you can plant that susceptible variety in the spring and do well, but you plant that variety in September in our area, you're gonna look at 50% loss. Then crop rotation is kind of the same thing. Just don't plant as much lettuce if you can, or keep it fallow. That definitely helps things. And then you can plant resistant varieties. That also can help you. So that's basically the management, and it's nothing new here. Now, and if I close this part up here, I do want to show you a couple things, and that's why we have this however. There's some bad news with those two tried and true IPM methods, and I think some of you have heard Tom's talk and some of the other things on this. There's some limitations with these two things. Crop rotation and planting resistant varieties, it's not a fix either, unfortunately. And some recent work has shown that if you plant other non-lettuce crops, the fungus is still growing on it. It's still growing on it. We have a big rotation with broccoli, cauliflower, spinach in Salinas, and Tom Gordon has shown that the Fusarium on lettuce grows quite well on lettuce, on the roots. The lettuce pathogen grows quite well on the roots of broccoli, cauliflower, spinach, and it's reproducing. So it shows that even though you're rotating with a non-lettuce crop, the fungus is doing pretty well on the roots of that non-host. And that's kind of contrary to the traditional teaching. We usually teach that you rotate with a non-host, the fungus is going to decline, it's all good. Well, it's more complicated than that. Now, to be clear, let me make sure this is clear here for everybody. The lettuce fusarium that's growing on the roots of these other crops, it's not causing disease. It's not pathogenic. It is only going to go to lettuce as a pathogen. But it's surviving on the roots of other plants, and it's enabling itself to maintain populations while it waits for the next lettuce.
Are the populations increasing, or are they just maintaining with those other crops?
Good question. He asked about whether the populations are increasing or maintaining. I believe if we look at the research, I think it was spinach. Spinach is the one where there's a significant increase. It's kind of really mind-blowing. We didn't expect that. And I think even on broccoli, cauliflower, there's a slight increase. So again, it complicates the picture. Now, this doesn't change anything in terms of practical day-to-day management, because you have to rotate crops, right? You have to rotate. If nothing else, for your product line and limited ground, you're going to grow something else there. So we're not saying don't grow something else, don't grow melons or don't grow broccoli. We're not saying that, but we're saying be aware that this is not the fix for Fusarium on lettuce, because this pathogen is versatile enough to grow on the non-host, which is a fairly new finding for us. You have a question?
What about down here when you rotate into a cereal? Are you seeing it on cereal roots?
Okay, the question about rotating to cereals. So we know cereals supports a lot of pathogens that are not pathogenic on it. So Fusarium fits in that. But specifically, this study that Tom Gordon did not include cereals, I believe. So specifically, we don't know. But now we're really suspicious because, if it's going to go on broccoli, why wouldn't it grow on rye too, or wheat or oat? So, so specifically, I don't know the answer to that, but I would say it's suspicious that Fusarium probably can at least grow on the roots of barley, say. Excellent question. And then the second part's the same thing. It's more bad news, but for the resistant varieties, for you companies developing resistant varieties, It's all good. We need those. We have to continue with that. But it's not a 100% fix because the Fusarium that's pathogenic on lettuce will grow on the roots of the resistant variety. Very clearly it does that. And in fact, on some of the resistant types, the population it goes sky high on that variety, but causes no disease because the variety is resistant. Yeah.
Do we know if it will reproduce on alfalfa roots if somebody went to that rotation for
Sure.
2 or 3 years?
Great question. The question about alfalfa is a rotation thing. How does it fit in this bad news picture here? We don't know. And this is some of these questions are really great ones because maybe that's part of the objectives of some research that could be done down here. You know, rotate with what you rotate. So we did with what we rotate with, and we got bad news out of that. So you guys should do the same study, but put in alfalfa, melons, barley, whatever it is, and see what happens. So really good point. So I do not know though. Yeah. Okay. Okay. A couple more minutes, and then I'm done. Okay. So this is the bottom line. This is the Slinski Valley way. You know, we think lettuce rotation, or I'm sorry, we think a crop rotation is lettuce, spinach, broccoli, lettuce. That's a rotation, right, for Slinski Valley? Yeah. Or maybe we're real generous and do 3 lettuces, and then we'll skip 1 year and then do 3 more lettuces. That's crop rotation. Well, that's part of the problem. We plant too much lettuce, back-to-back lettuce. Lettuce everywhere, that's Slinski Valley. That's why we have so many problems with the soil-borne issues, because of the dynamics we talked about. Right? Okay, then I'll finish up with this. Just a word about detection and Fusarium and trying to find Fusarium in the soil. It's very complicated, and I know you've had other speakers address this, so let me just give you our perspective on it. It's difficult, complicated work, and it's not easy. So what we're trying to find is the specific Fusarium that goes to lettuce in a bag of soil, right, in a field. And it's not that straightforward. So to illustrate that, here's a schematic. So that blue box, that's all the Fusariums in soil in a particular case, okay? So there's maybe 20 species of Fusarium in there. And maybe 4 or 5 predominate. We're only interested in the oxysporums because the oxysporum includes the group that are pathogenic, and therefore this includes the group of the lettuce Fusarium, okay? But there's a big box of Fusariums, and we're looking only for one group, the oxysporum. But let me show you the oxysporum group. I'm going to blow this one up. Even that is not simple because there are a ton of oxysporums in the soil. The pink is all the oxysporums in the soil, and most of them are not pathogenic to lettuce. In fact, most of them are not pathogenic to anything. They're kind of in there as saprophytes, natural occurring organisms that have some ecological purpose, but they don't infect lettuce or anything else. The ones that do infect lettuce and do infect other crops, it's a very small triangle in that whole picture, okay? So when you put this all together, let me point out first. So let's blow this up. What do we mean by the oxysporums? And I think you know this. The Fusarium wilt pathogens are almost always an oxysporum group fungus, okay? That's why we're concerned with oxysporum. Lettuce Fusarium, strawberry Fusarium, celery, it's all the oxysporum group. And because they are only going to one host, we give it a special designation, this FSP, Forma specialis. That means that pretty much that fungus only goes to one plant. So the one on lettuce is FSP lactuci, only goes to lettuce. The one on celery, FSP api, only celery, etc. So it's very specific. These are very specific. But you put it all together, we're trying to, and others are trying to find just the one Fusarium that goes to lettuce in this whole big mix. And it's not an easy task. And we don't believe there's a good method that is foolproof, that accurately tells you which ones go to lettuce in this bag of soil, and also how much is in there. We do not believe the technology is accurate enough to give you that information. There's some fantastic tools out there that may help us down that road, but we don't feel that there's evidence that that is being done really thoroughly. And so I just want to close by saying we are all trying to quantify, for you and for us, how much lettuce pathogen is in the soil. And we have really a tough task because it's a small target, because of this picture I've shown you. You need a specific test just for that one type of Forma specialis that only goes to lettuce. There's a lot of genetic overlap with all these oxysporums. You know, that's the problem. There's a lot of overlap in how their DNA is lined up, and so you may not have a really good tool to separate those reliably over time. And then even if you could find it, once you find it, we don't have a threshold, meaning, so I give you a number, this is how much Fusarium oxysporum from lettuce is in your soil, and I give you a number. What does that mean? I would propose to you, we don't know what it means yet. We don't have a threshold where we could say, well, if it's above this line, that means you can expect 50% crop loss. If it's below this line, you're going to have maybe 5% crop loss. We don't have those thresholds. And so I would say that's another area of research where we maybe can collaborate. Frank Martin is going to talk about some of those things, I think. But the whole thing about detection and quantifying, it's a big mess right now, in my opinion, for lettuce. We cannot have, we don't have the information and technology to hone in specifically to indicate those two things. All right. I think I'm done with that. Again, I'll be here for the whole session and I guess the panel afterwards, so we'll hold questions till then, right?
Frank Martin is a research plant pathologist at USDA-ARS in Salinas. Before that, he was at the University of Florida, and his current research interests include investigations on the ecology, biology, biological control, and genetics of fungal soil-borne pathogens and other rhizosphere microflora. And his current responsibilities with USDA is the development of alternatives to methyl bromide for the strawberry and vegetable industry, and the development of molecular markers for detection of pathogens controlled by methyl bromide.
As Steve pointed out, that it's a real challenge working with Fusarium oxysporums because they look morphologically similar when you look at the spore structures. And if you're someone like Steve or Tom Gordon that has looked at a lot of cultures from a specific area, they could probably have a pretty good idea of which one is a forma specialis lactucae and which one isn't. But for the rest of us mere mortals, it's a little bit more difficult to just look at something like this and have a general idea of saying definitively, yes, this is lactucae. Because your Fusarium oxysporums can be very aggressive saprophytes in the soil. They can colonize a lot of the root tissue. They can colonize other organic matter in the soil and reproduce and propagate. And one of the challenges is, as Steve mentioned, you can go in on the lettuce roots and you can pull out Fusarium oxysporums that are not pathogenic on lettuce. And by that we mean it doesn't go systemically. It doesn't get inside the plant, move throughout it, and kill it. It can colonize some of those cortical cells, and it can reproduce and maintain population structures, or as Steve mentioned, in some cases, increase, but not cause the wilt disease. And actually in some crops, tomato, chickpea, they actually find there are some strains of Fusarium oxysporum that can systemically colonize the plant. You can go up into a petiole and take the petiole and recover that particular strain. Yet in pathogenicity tests, it causes no disease. So it's a little bit of a complex system to try to figure out what's going on with these. And in many examples, they find that the evolution to cause disease on a particular host has happened in different places. So you have organisms that, like on Forma specialis cubensis on banana, you can have organisms that are very evolutionarily distinct, causing disease because they've independently evolved to cause that type of disease. So this is what makes it so challenging in working with the Fusarium oxysporums. I've done most of my career has been working on oomycetes, and they're diploid. I used to think this was great now to work on a haploid organism, trying to do genetics and molecular work, and I'm not quite sure that is any easier with all of this complexity and trying to figure out what's going on with the oxysporums. So as I mentioned, they can be difficult to identify. You can have isolates of non-pathogenic, and this is what makes it a little bit more challenging then to go through soil samples and to try to quantify what's in the soil, as well as taking a look at recovery from plants to identify what they are. And there's, as Steve mentioned, there's the 4 races here, and currently you use host differentials to separate those races. Well, it's really difficult to have to go through all that. It'd be nice if there's some molecular means to go through and do that classification. It'd be nice if there were some consistent molecular tools that we could come up with, and we know specifically it is only Forma specialis lactucae. Now we're just starting our work with lactucae and looking at other saprophytes, but we've done a lot of work with Forma specialis fragaria on strawberry. The standard parts of the genome that people sequence to identify Fusarium oxysporums to Forma specialis, we find isolates that are completely non-pathogenic on strawberry are identical to those that are pathogenic on strawberry. We have a postdoc, Alyssa Burkhart, who did a lot of the work that I'll be talking about in a minute. She's now working with Steve. She thought we had everything all straightened out until we found that there were a lot of these isolates that were non-pathogenic that had the exact same sequence analysis. And she had all of our greenhouses filled with plants doing pathogenicity tests. So this is another layer of complexity that comes on. And what we're trying to do then is develop some of the molecular resources that we can use to take a look at this in a broader sense. How can we tie this down to, is this Formis specialis lactucae? What race is it? And then also to take a look at, are we looking, these things don't have a sexual reproductive stage. They're clonally reproducing. Are we finding that the isolates that are in California and in Arizona, are they identical? Have there been multiple introductions of different genetic backgrounds of the race 1 that are present here in the United States or other parts of the world? So is there a way that we can kind of come up and genotype our isolates a lot more quickly and efficiently to determine exactly what we have? And our approach is, rather than taking a look at the literature and seeing if people looked at this piece of the DNA or that gene, our approach is to kind of step back and say, let's sequence the whole thing. Let's use some of these new DNA sequencing technologies to generate a large data set by sequencing the entire genome, and then use computers to go ahead and figure out the answer for us. So there's a new technology, but it's not new now, it's called Illumina, but we're at a point now where we can send out 25 isolates for sequencing, and it might cost us $6,000 for those 25 isolates, but we get tons of data that we can now work with this. On some of these runs, we might get 600 million snippets of DNA that are 150 base pairs long, which is an awful lot of data. And then we use a computer that's got an awful lot of RAM and storage space, and we go ahead and do our analysis with that computer bioinformatically to help clarify exactly what are the similarities and the differences among some of these isolates. So we've sequenced race 1 isolates, 8 from in California, 2 from Arizona, 1 from Japan, and 3 from Europe. We've had collaborators in Japan and Europe that have sent us the DNA to work with. The race 2 from Japan and 3, and then race 4 is, as Steve said, the Netherlands, Belgium area. And we currently have a whole series of isolates that Tom Gordon has been working with that he believes are non-pathogenic on lettuce. He's got the path test going right now. Hopefully in the next week or 2 he'll be able to give us some results. But we're setting up to sequence, I think it's 13 or 15 isolates, because it's important for us to know what the sequences are for those that we're recovering from lettuce, but don't systemically colonize the plant and actually cause disease. So it's an important part of why we're trying to do this. So what are we going to do with this information? Again, we want to understand the evolutionary relationships of different isolates of the Formis specialis lactucae. Can we take a look at these races? Are they closely related? And if they are, then maybe we can develop a one marker system that's going to work on all 4 races. If they're not, then we might have to approach this in a different way. What about genotyping isolates? What we designed for race 1 for California and Arizona, is it going to work for the other isolates in the event they, at some point in time, get introduced? But we also want to use this data then to do the diagnostics and develop markers that are going to be specific for the particular Formis specialis that we're interested in. So again, with the evolutionary relationships, we've got the 4 races, and we want to compare that to the rest of the Fusarium oxysperms that we have data to get that broader picture. And this is going to help us, again, understand when we design our markers, is one marker going to work for multiple races? But also it's going to help us identify, make sure we include specific Formis specialis in our screening assays for specificity, because now we know phylogenetically or evolutionarily what is the most closely related taxa. And then we want to make sure that, since that probably has a higher chance of having sequence similarity, that that is included in our assays. And if we don't have that DNA, we need to start working with people that do have it so we can get it to include in the trials. So we've been collaborating with Dave Geiser at Penn State. Dave runs the Fusarium culture collection there. He's one of the experts in the world in working with Fusaria, and he has an awful lot of Fusarium oxysperms. Many of the ones that he has worked with that we have sequenced, we know for a fact that they're pathogens. They just haven't been recovered from strawberry. We know that they actually cause disease on strawberry, et cetera. And he's got a 41 gene set of data here that he's gone through on oxysperms that they've assembled the entire genome. He knows that those genes are distributed on different chromosomes, because we don't want to get something that's all in the same place. We want to take a look at evolution in different parts of the genome. And he's found that these 41 genes, totaling about 69,000 base pairs, are very good for determining phylogenetic relationships among all the Fusarium oxysperums that he's been working with. And so if we take a look at just our lactucae and we reroute our phylogram to the form Specialis fragarii off of strawberry, we find that there's very little variation in these 69,000 base pairs among the race 1 isolates. And interesting, the race 4 is very, very similar. There's only a couple of base pairs difference out of that 69,000 that are separating the race 4 from the rest of the race 1. But our race 2 and our race 3 are a little bit more distantly related. So this is just looking at the lactucae, but it's important we know what this means in the broader sense for all of the Fusarium oxysperum. And so these are, I don't expect you to read all of these. These are a wide range of Fusarium oxysperums that we've been sequencing for Dave Geiser. And then we extracted the DNA of these 69,000 base pairs. And we can see again our race 1 and our race 4 is clustered on the bottom, but our race 3 and our race 2 are distinctly separate. So as I mentioned earlier, the form Specialis cubensis have phylogenetically distinct lineages that have independently evolved to become pathogens on banana. The same thing has happened with lettuce. These are distinctly separate into different groups there. So it kind of means our chances of trying to design a marker system that's going to identify all 4 races just isn't going to work. It might work for the 1 and the 4 because they're so closely related here with just a couple of base pairs difference. So one of the approaches that we're doing is, again, not just on lactucae, but we have over 240 isolates representing 47 forma specialis. Now that sounds like a lot. My understanding is that there is about 104 or 106 recognized forma specialis for Fusarium oxysperum. So we have a good start. We're not there yet. This 240 isolates includes isolates that are saprophytic on the host that they were recovered on and do not cause disease. And so that's why it's important. We've done this with strawberry. We analyzed a broad number of isolates that were non-pathogenic on strawberry to make sure that we were including their sequence data when we were screening for developing molecular markers. Our raw sequence data is over 5 terabytes. So we have quite a bit in there, and a software pipeline has been developed to go through and identify unique sequences that are only going to be present in, say, the race 1 of form Specialis lactucae. We screen it against the rest of that 5 terabyte data, making sure that that sequence is unique. And then we use those as a preliminary screen bioinformatically on the computer. Now we design primers to go ahead and amplify that with DNA to make sure that, in fact, they are specific. Now I should note, we just got funding from a California Department of Food and Agriculture Specialty Crop Grant for 2018 to go ahead and expand this database. Somewhere between 60 and 90 additional isolates will be sequenced. We're going to develop a database that will be available to others that has all of this information in it. We're trying to streamline the software so computationally it's a lot more efficient and make it a lot easier then to go ahead and pull out some of these unique sequences that we're looking for. Because our approach on fragaria, developing markers for that work, I think it should work for the lactucae, and I think the approach should work for a number of other forma specialis. So we're just trying to develop a systematic approach for doing this so other people dealing with Fusarium problems can have these tools available. Then we use a comparative genomics approach there in that software thing to go ahead and identify unique sequences. Now this approach has been used for the Macrophromina phaseolinea. Alyssa Burkhardt, again the postdoc who did it for the Macrophromina and the forma specialis fragariae, now works with Steve. And Steve was heavily involved in validating and getting this whole thing set up and helping us make sure that it's actually working in the field. So this type of approach on Macrophromina was all done by hand, and it was very labor intensive. I spent probably a week in front of the computer doing that. And Alyssa designed this software pipeline that we then used for the form Specialis fragariae, and it worked far more effectively and efficient. We've done a lot more fine-tuning on it now to make it more computationally effective. So what we've come with on the race 1, out of all that database, we have 540 fragments of DNA that we believe are unique. We've gone through and screened for race 1 and race 4, and even though they're closely related, we only have 11 fragments that seem to be unique to those 2 races. We don't find anything that has all of the races. which doesn't surprise me when we take a look at that phylogenetic grouping that showed how distinctly different that they were. Now, since race 1 is the one that's most present, that's what we've been focusing our attention on. If race 4 happens to come along, that's great. What we found is with further testing, when we took a look at more isolates, only 4 of the 11 targets were conserved in all isolates of the race 1 and the race 4. So this is one reason why I think we're just going to drop back and focus on the race 1 and see if we can get that going first and have different markers for the race 4, 2, and 3. And we're waiting for the sequence data from these non-pathogens. We're going to retest these isolates, and then we're going to get into designing primers in the lab, doing the wet work to evaluate how specific these markers are. We have found in the past, in working in the form Specialis frugariae, that not including data like this in here wasted a lot of time. A lot of time was spent developing a marker system, and then when we expanded the isolates we tested it on, we found out it wasn't as specific as needed, and we had to go back to the drawing board. So right now, we're just going to wait for these additional isolates to get sequenced. We're going to retest the markers that we've already come up with, and then move forward with the wet lab to see what we can do to come up with some of those markers. So 2 different types of assays we're looking at developing. One is a TACMAN assay. This is what we call real-time PCR, meaning that you can sit there on a computer screen and you can watch amplification because it generates a fluorescent marker. The concentration of the marker is directly related to the amount of DNA that's in the sample and is being amplified. The TACMAN system works quite well. It's very specific. It's highly sensitive. The one downfall is you have to do formal DNA extractions. And sometimes we find with soils, we have to be careful on how we do the extractions because compounds can come out with the soil that actually inhibit that amplification. So there's procedures we have to include in there to make sure that that isn't happening from our DNA extractions. And we have a quantification assay currently for Verticillium dahliae that's accurate to 1 to 2 microsclerotia per gram soil, and for Macrophamina phaseolina, 2 to 3 microsclerotia per gram soil. And this again, Lissa Burkhardt did a great job in getting the Macrophamina set up, and a prior postdoc, Guillaume Billedou, did a great job on the Verticillium. We're currently working on this on Forma Specialis frugariae. The strawberry industry, like yourself, is looking for a way to be able to rapidly quantify the amount of the pathogen in the soil. So then we can take a look at risk factors as far as are there differences in cultivars that can be planted, other choices that can be made up front. I think the last number that I saw, the cost of production of strawberries in our neck of the woods is about $61,000 per acre per year. So the growers want to make sure that they don't run into problems with disease and lose their shirt, because it's a very expensive crop to grow. All right, the other one is this isothermal detection technique. And this is a really cool system because you only have one temperature. Doing PCR, you have to spend $25,000, $30,000 on a machine that ramps temperatures up and down and helps read it. You have to have a service contract. It costs a couple of bucks every time you run a sample. You have to spend a couple of bucks every DNA extraction. It can get rather expensive. So what happens with this one, it all works at a single temperature, and it's done by a company in the UK. Agdia has purchased the rights to it, and they have some tests that they are selling and kits that they have. And what's great is it's a system very similar to real-time PCR. You just have longer primers and probe, and it's a different type of system because it's a recombinant polymerase amplification. There's several enzymes that are involved in there that bind the primers and the probe to it, unwind your DNA, and then put it where it's supposed to go. It's a little bit more complicated, but what's great is you don't need to do DNA extractions. You can take a field unit that is made by a company in Hawaii. It's about 3 times the size of a pack of cigarettes. You can have a Bluetooth connection on your smartphone and run it off of that. And within 15 minutes in the field with a plant sample, you can tell if your pathogen is present or not. Now, the TACMAN's a little bit more sensitive, but for the fact of rapid field diagnostics, it works very well. We've been very impressed with it. We even turned Steve into a molecular biologist when he was with UC.
You were going to say that in public, though.
I'm sorry.
My image.
And so it's something that's very simple and straightforward to use. These are some of the pathogens that we already have set up on strawberry that have been used by UC Cooperative Extension, and there's several other commercial diagnostic companies that are using these as well. We have one for Pythium species and Rhizoctonia, working with a Pryor postdoc who just moved to Michigan State, Tim Miles. That work, we're in the process of final validations, but we have it all set up. There's just a few things we have to finish up. So those tests are already available. The downside is you don't have a kit that you can buy to do it all. We have been working with the company. TwistDX is owned by a medical diagnostics company. They see the money working in the medical field, not in the ag field. And we've been having an uphill struggle trying to get them to formulate kits with our primers and probes in it. They're looking at what it takes for product development and what sales will be. If things don't work out with AgDia or with TwistDX, there's always AgDia, because that is what their wheelhouse is and what they try to develop. So we currently have a Phytophthora kit that is they formulated for us, and it's out in testing in several diagnostic labs across the country in an effort to try to get back to the company to say, this is what we need to do. Because right now it's not simple. If you have a lab with someone with some technical experience, it's simple. But if you don't have that facility, if you don't have the ability of people to go ahead and make dilutions, the primers and probes alone cost about $1,000. If you don't have the ability to go ahead and do the dilutions, test them, have DNA samples, it's not going to work for you. But if you do have that ability, it works very quickly in the lab to do diagnostics. Steve can fill you in on how it's working in his program. But our hope is that we can get this thing set up, that these types of assays are available to the general public. Now, as I mentioned, one of the things that we want to know is, are all the isolates that are race 1 that are present here in California, Arizona the same? Are the race 1 isolates in Europe and Japan and Australia, are they the same as what we have here? Right now, there's no genotyping ability out there that we've seen. So what we tried to do is use this genomic data as a way then to mine it for specific information that we can use. So we have the assemblies of the Formis specialis lactucae isolates, and we've gone through and identified regions that are highly conserved in all of the isolates that we have looked at. And then we've gone through and looked at all pairwise comparisons and seen what are the differences among these. I should say that if you have any other isolates that you think are growing a little weird or a little interesting or a little different, I'm happy to sequence them. So we have 37 pieces of DNA totaling 7.26 million base pairs. And we can go through now and take all of the data from the genomic sequencing of the different isolates and put those reads onto those and determine how many differences are present. And these differences can be things on what we call single nucleotide polymorphisms. Rather than being an A, is it now a T or a G or a C? Those are pretty simple mutations. Or we can have simple sequence repeats. This is if you've ever watched a police show and they talk about, oh, this person did it because we tested his blood, those are simple sequence repeats. It's where you might have a CAT, CAT, CAT multiple times. And when DNA replicates, it kind of gets confused because there's so many of them and it slips. It's like, for myself, I have some of the microsatellites that my mother has, my father has, but I also have some that are unique to me, which make my microsatellite profile different from my brother's. So we're taking a look at these bioinformatically, and then we have things where there might be a base added in one isolate and absent in another, and that's called an indo. We're still doing some of the data analysis in a broader sense, but what we've seen, if we go back to this phylogenetic tree here, we take a look at the race 1 isolates, there's only 5 to 10 base pair changes out of that 7.26 million base pairs. It's pretty much a solid clonal population. We're not seeing a whole lot of variation in the ones from Europe, California, or Arizona, or Japan. We have the one down here, though, with race 4, and that has 211 of those base changes, 10 differences in the simple sequence repeats, and 26 places where there has been pieces of DNA added or taken away. So it's a little bit more variable, even though there's only 1 to 2 base pair differences in those particular genes we used in the phylogenetic analysis. From a genome-wide analysis, there's a lot more variation. But when we get up here to race 2 and 3, the differences are just astronomical, which is what we would expect, because phylogenetically, they are completely distinct entities from different parts of the phylogenetic tree. So this is something that we're trying to do to evaluate what's going on with some of the isolates. And again, I'm happy to look at additional isolates, and I'm looking forward to some of the ones that Stephanie's going to be able to provide to make sure that we can take a look and account for all the variation that's out there. Now, there was a paper that came out recently using, saying that they have a diagnostic assay for Formis specialis lactucae using what's called loop-mediated isothermal amplification. Similar to that RPA assay I spoke about, it's done at one temperature, but it's a completely different approach in chemistry. The problem is on this paper, they don't give you any specifics. There's no primer sequences, there's nothing. All they do is they say, this is what we did and it works. But they did say what region they took the sample from. So it's a 599 base pair fragment that was pulled out in a prior publication. And they validated it against 21 forma specialis of oxysprom. I should say I've been emailing these folks asking specifics and I haven't gotten any responses yet. So what we did, now since we have all of this data for 240 isolates we have sequence data for, we can go ahead and take that target sequence, that 599 base pair sequence from our race 1 isolate and pull it out using that published sequence. So now we have the one for the lactuque. We can take areas on either side of it because our analysis, we need to have that piece of DNA a certain size. And then we ran it through our pipeline looking to see, is this truly a unique sequence? And unfortunately, what we found is it isn't. None of these were included in their survey that they went through and tested for specificity. Now, I haven't tested these because I don't have any of the information, but these results show that they're almost identical. There's only 4 or 5 bases different, and those differences are random throughout all of the sequence. So I'd be very surprised if their diagnostic assay is going to be absolutely specific for lactuque, strictly based on this data. I don't know that for a fact, but looking at the bioinformatics, I question it. And I hopefully at some point in time we can find out more on what the specifics are in that assay so we can test it. Because if they already have something worked out, I don't want to reinvent the wheel. It'd be great if it works. So just to try to wrap this up here with the phylogenetic analysis, we can see that the race 1 and the race 4 are very closely related. Race 2 and the race 3 are very distantly related. They're completely phylogenetically distinct, those 3 groupings, into the 3 major phylogenetic clades. We're working now with Dave Geiser to take a look at a larger number of isolates to include it and come up with a little bit more comprehensive phylogenetic analysis of where these things specifically are. We are going through and identifying regions that are specific to unique sequences. We're looking for these to see how well they work for diagnostic markers for identification of the different races. Focus first is race 1, but then we'll hit the other 3 races because who knows, at some point in time something might be introduced and we want to have the tools available and ready. We're also working with other collaborators in Europe trying to get additional isolates of race 1 and race 4 so we can get a broader geographic distribution compared to the ones we got from the folks in Italy. We're also going through in genotyping using large data set of 7.26 million base pairs, which gives us a pretty good estimation then of genotypic differences among these. We're going to go through the race 2 and the race 3 and do the same type of thing and structure it so we can get good genotypic data for these 4 races. So again, in the event, if there is an introduction, if there's differences among these genotypes, we can say, this is where it came from. Let's go back and take a look at how it might have gotten here, what channels it came to come through here. And then mainly, I just want to highlight the importance of the sequence database. Again, this is something we want to set up. Tom Gordon's collaborating on it, and Dave Geiser. We want to get this set up to get a comprehensive data set as well as the software that people can use for other people. We want to get this validated with our forma specialis lactuque. We're also working with people that are interested in API and like a Persici race 3. And once we get those things set up, hopefully, and validated, we'll have that out for everybody else to have it available to them. And so I'd just like to thank our collaborators here. And Stephanie is a new collaborator on this, helping us out with isolates. We're interested in getting pathogens as well as non-pathogens that are recovered off of the roots. And she has an arrangement now with Tom to take a look at some of these things. And I should mention, as somebody did earlier, Steve Koike was very instrumental in helping us get the forma specialis fragariae and the macrophamina assay and the verticillium assay up and going. These are the folks that provided the DNA for us, and the California Leafy Greens Board was very instrumental in providing funding. And we do have that CDFA grant now to expand it. So I think that's pretty much it. So the question is, have we thought about looking at specific regions of the genome that might be associated with that response between the host and the pathogen? I can think of probably a dozen labs and millions of dollars being spent right now that are doing exactly that. And there's one series of genes, they're called 6 genes, SIX. secreted in xylem. They're very small, cysteine-rich proteins, and they find that they are associated with virulence in some of the Formis specialis. And Formis specialis conglutens on cabbage, and race 4 of the Formis specialis cubensis on banana. They've gone on and they've mutated that gene and find that the virulence on the host has gone way down. So this is an area that is being looked at a lot more now. I don't think it's to the point where I want to put all my eggs in one basket. It's certainly something we're looking at, but it's an intriguing possibility. I think there are multiple 6 genes that are out there. And I think there needs to be a lot more work to really clarify the specific involvement in each of them in virulence. But it is an intriguing possibility, and I think in the next couple of years we're going to see a lot more interesting data come out of that. And that will certainly simplify things. But one of the questions is, are we going to see enough differences in some of those genes that we can use for developing a diagnostic marker? Because we're looking for something that can be done rapidly. I mean, I can sequence something and give you definitive stuff with the data we have, but you're not getting the result today, tomorrow, or the next week. So we're looking for things that can be quick. But that was a very good question, and it's right on target. And as I said, there's at least a dozen labs and millions of dollars a year being spent on trying to address that very question.
And I have to admit, I failed miserably with a bio for Barry. Sorry.
They work at the same university.
University of Arizona. We just met in person today, but I've spoken to him several times on the phone. Done quite a bit of research on Fusarium Wilt of lettuce. Looked at infection of seed, and he's doing some projects that with us at YCEDA.
Okay, so it was kind of interesting. I started working with Fusarium wilt of lettuce in 2001 when Mike first found Fusarium wilt here. And we started off working with seed, and we've alluded to that a little bit here. And what we did find, indeed, that seed, we looked at over, or almost 100 lots of seed used over 2 to 3 years here in the Yuma area, and we did not find any Fusarium wilt pathogen in the seed. And then we did some studies in the greenhouse to examine how that pathogen might move up into the seed. We infected some lettuce plant, and we followed the recovery of the pathogen from the roots to the stem to the inflorescence and up into the seed, and we found that in fact it was very difficult for the pathogen to move all the way up into the inflorescence. But what we did find is that we could easily contaminate seed during the threshing process. In other words, if we mixed infected lettuce, if infected lettuce material was mixed with clean seed during the threshing process, you easily could infect seed. So there's discussions about how resistant cultivars and resistant plants can still harbor the pathogen just is very important, because usually when people go do seed production and they look, should I harvest seed? Is there a problem with contamination? They usually look to see if they see symptoms in the field. And so in this case, if you're looking at it and you see symptomless lettuce plants and you think, oh, my seed is going to be clean, it in fact might not if in fact the lettuce plant harbored the pathogen in the roots or in the stem. So you have to be very careful. And there you probably would have to test the soil and only grow your lettuce seed crop in soil that has been determined to be free of the pathogen. We talk about how the pathogen can move from area to area to area. It's probably not on seed, but let's not forget, it probably moved across the oceans on seed, not on infected soil. So seed-borne pathogens is still a problem. Now what I'm going to do today is talk about not some of our early work on Fusarium wilt of lettuce, but some of the more recent studies, starting with our 2006 Specialty Block Grant Program grant from Arizona Department of Agriculture. And this was with Paul, myself, Mike Matheron, and Murat Kassir, who's an engineer in the Biosystems Engineering Program at University of Arizona. Again, so we had a couple different, and I'm going to go through this really quickly. Paul Yoli gave me a few minutes, and Paul knows I'm very long-winded, so I'll just take the time that's necessary and talk about the disease. Okay, so we had a number of objectives in our 2016 Specialty Block Grant, and we started out looking at cultivars and chemical and biological protection for product evaluation, and Mike was the one who was doing most of this work, and so you'll have to ask Mike the questions involve that. But I wanted to talk about some of our work on detecting the pathogen. And I think this was really great having both Steve and Frank here, because we work together on this, and a lot of our work will cross and complement each other and build upon other people's findings. But we also wanted to look at rapid disease detection using these novel DNA methods, these isothermal DNA amplification methods, particularly the recombinant polymerase amplification methods and the loop-mediated isothermal amplification methods. And Frank mentioned both of these as well. We also wanted to add onto this mix the possibility of using remote disease detection, using multispectral imaging, perhaps with drones flying over the fields or something. We wanted to do some preliminary work into this area. There's been some interest in looking at plant health using multispectral analysis, and we wanted to also look at, can we use these techniques, these technologies, to evaluate disease pressure or the incidence of disease as well. So this is our experimental design. It's kind of an elaborate experimental design. We wanted to look at the impact, particularly with the multispectral analysis. Can we differentiate using these different spectra stress on lettuce plants from Fusarium oxysprum, Sclerotinia, or maybe even water stress? We wanted to see, can we differentiate those signatures of stress based upon those three challenges? And let's see, sampling protocol, we looked at symptom development. Of course, we know what the symptom development of water stress is, right? It's wilt. But we wanted to look at symptom development of the pathogen infection, whether we got necrosis, whether it was vascular necrosis, stem decay, crown decay. And then, of course, we're going to do image capture based upon our multispectral analysis. Let's see, we use randomized complete block design, all this standard techniques for doing proper statistical analysis. Here's our experimental design. It's rather elaborate with many different water treatments, pathogen treatments, Sclerotinia and Fusarium. And let's see, this is Dr. Murat Kassir right here looking at some of our plants, our greenhouse studies. And for our detection of the DNA, of the pathogen DNA in the lettuce, we compared it with a standard nested PCR technique that we developed early on in the detection of the pathogen in seed. And this nested PCR technique is tried and true on many different pathogen groups, whether you're dealing with verticillium or Fusarium or Macrophamina. And then we wanted to compare it with the development of this recombinant polymerase amplification product using the AgDIA platform, okay? And again, it's an isothermal reaction with an enzyme-linked antibody detection. It's very sensitive. It's kind of like any of those medical diagnostic test strips that you might use for a number of different diseases. And then we also compared it with this LAMP technology. Again, it's an isothermal reaction, very fast reaction, and this was a product put out by New England Biolabs. Each method is highly specific and highly sensitive, right? But each has unique challenges, characteristics, values, and we wanted to compare them in a single study. Let's see, and as Frank alluded to, this is the challenge here with both the RPA technology or the LAMP technology. The primer development, the antibody development, the probe development is very elaborate. Much more difficult than using a standard nested PCR reaction. And so it's time consuming. It's an iterative process. And unless you have something like Frank has, which has the whole genome, and you can go in and do an in silico prediction, which we did to an extent. We used some of their primer design platforms that they promote in both of these companies. But again, it's a trial and error kind of experiment. And as Frank mentioned, primers can be very expensive for some of these products. So, for example, in the New England system, they said, no, you need to, and there's many primers involved, you need to predict maybe 6 or 8 primer sets and then test them all and see how they do. Well, that can be very very expensive. Okay, I'm just going to go right through these real quick because I don't want to show those. This is the multi-spectrum analysis that we did. And I thought this was very interesting. Again, Dr. Murat Kasira is the engineer here, and I'm not going to pretend I know much about multispectral analysis. And I have to sort of rely on Dr. Kasira to help me through and walk me through. But this is really what we're looking for. We're looking at a full spectrum analysis of reflectance from these lettuce plants stressed in different ways, and then we're going to look at particular spectra, the blue, the green, the red, the near infrared, and see if we can see signatures of stress with these different stresses that we applied, and see if any one of them appeared unique to the others, okay? And this is used in a lot of different purposes for looking at, detecting morphology or other kinds of stresses of the plant. And here's our multispectral analysis chamber. Again, we put 3 plants down there. Here's our camera mounted up at the top here. And we would image these plants throughout the growing season, throughout the, I think it was a 5-week growth. Okay. Now, I mentioned we had 2 trials. We usually, whenever you do an experiment, you always repeat it. And we had our first trial, repeated the second, but we had a challenge in those first 2 trials. In that, in this greenhouse study, we actually got very little symptom development, which was problematic because when we've done Fusarium Wilt studies in the past, we had very good symptom development. So we decided, and we were using a type of a soil in our first studies that was a standard greenhouse potting mix. We thought this would be appropriate. Little symptom development. We said, okay, so we need to try to promote plant disease. We always do that as plant pathologists. We try to promote disease. And so we mixed some soil. We had a couple different soil mixtures in this second experiment and challenged the lettuce with several different strains of Fusarium oxysprum lactucae. Again, in order to get expression. And here's some of the results of our multispectrum analysis. I think I'm probably. Oh, okay. I thought, okay. Can you guys hear me okay? I can still call. So here's some of the results that Murat Kasira got with his analysis. And some of these indices they use, and I can't really explain a lot what it is. This is the normalized different vegetation index. This is a common one used to look at crop canopy. There's things like near infrared reflectance down in here. But what he did, the take-home message here, is that he really found he was able to detect stress. Yes, he was. And he was able to detect it in the pathogens without water stress. But he found no difference, at least to date, in stress caused by drought or stress caused by pathogens. And I say no differences to date because these multispectrum analyses are very data-dense sets. In other words, it's sort of like some of these bioinformatics data sets where Frank is looking at 1 million different reads, 600 million reads. With this multispectral analysis, it's the same kind of thing. You look at a lot of different data points and you can go back months later and look at them in a new way and evaluate. So Dr. Kasira is still looking at this and seeing if he can somehow tease out some differences. But to date, we have not been able to find differences between drought stress and pathogen stress. All right. There are symptoms. Now here's a challenge for us in this greenhouse study. In trial 1, no symptoms were observed. The plants all looked healthy. We did see necrosis in the roots, though. We did see, after 3 weeks, in our inoculated plants, we could see necrosis, but we didn't get symptoms, which was interesting because it showed that the pathogen was infecting a susceptible lettuce cultivar, but it wasn't showing symptoms. Okay? In trial 2, again, we only found 2 plants that showed symptoms, but again, we noticed necrosis in the plant roots. 3 weeks after inoculation, we were able to see necrosis. And in our trial 3, when we actually used some different soil types, we got a lot more more symptoms developed. And again, we found necrosis was both noted in the symptom plants and the symptomless plants. Now, and here's an example of some of the necrosis that was noted in some of these plants that showed no symptoms. The cultivar, even though it was a susceptible cultivar recommended by Mike, it was able, under the conditions we were growing it in the greenhouse, was able to maintain the plant integrity, not show symptoms, even though it was infected. Okay, and so here's some, here's some positive DNA samples. Again, this is the AgDia kit. It's really simple to use. Frank's used it. Many of you might have used it for other diagnostic purposes. I think there's one for Fusarium Wilt race 4 of cotton, and people have used it here in Arizona because we don't have race 4 in cotton, I believe. But our trial 3, and I'm just going to give you some examples of our trial 3, the ones where we varied the soil samples, our trial 3 results. 36 roots were tested using the AgDia kits. 18 of those were inoculated, 18 were controls. 10 positive out of the 18 inoculated. So we were able to pick it up, but not nearly at 100%. In other words, our primer design was not nearly what we wanted, or perhaps our methods of DNA isolation were not as exact as, or precise as we wanted to. Now, out of the other 18 tested, we got 4 false positives. So there we do have a problem with primer design. 4 false positives. But it's probably better to get a false positive than a false negative, maybe? Maybe. Using the NEB loop lamp technique, 36 roots were tested. 14 were positive out of the 18 inoculated. We're much better. We're getting better. And more importantly, no false positives. Okay, so we're making progress here. We haven't dialed it in. And probably it's going to be a combination of improved methods for DNA isolation from these infected lettuce plants and improved primer design as well. Now, okay, summary. Greenhouse symptoms were very poor. Necrosis was evident in the absence of symptoms. Multispectrum image analysis detected both the water stress, pathogen stress. We developed a protocol, a DNA protocol, where we could use it. I actually have some other data I want to show you here in a minute. We could use it both for the nested PCR, the LAMP method, and the RPA method. And let's see, what do I want to say here? Both the AgDia and the New England LAMP method performed well, but not as good as we'd like. So more work is necessary. I want to just jump, and that was our 2016 study. Now we've been doing this for a couple years now. Our 2017 study, this was funded by the Arizona Iceberg Lettuce Research Council, and we had a slightly different objectives for this study. In this, we were focused on detecting the pathogen in field soil, not in lettuce plants, in field soil. And we partnered with our company Trace Genomics here. And what Trace Genomics does is similar to what Frank was mentioning, where you're doing a whole genome analysis and you collect millions and millions of DNA reads, except Trace Genomics is doing it on a whole community, a soil community. So they're looking at both fragments of plant DNA in the soil. They're looking at all different pathogens DNA in the soil, the whole community. Again, collecting 600, maybe 1 billion reads, and then using bioinformatic techniques, using powerful computers to search through all those reads to find signatures, to find the DNA that you're looking for. It's sort of like a needle in a haystack, but the computers can do it much more effectively. And our objectives here were to conduct a hierarchical sampling. That means a very structured, stratified sampling of 3 lettuce fields with varying histories of Fusarium wilt. And then we were going to use conventional plating on petri dishes of that soil to see if we can get some numbers. Steve was mentioning it's hard to, when you get some of these, when you get some of this information from how much pathogen is in soil, it's hard to put that in perspective of, is this a problem or is it not a problem? If I get this sample, if I get this number on the field, is that going to be a problem? Is that going to be 50% loss, 25% loss? Well, we wanted to dial in a little bit more carefully by actually getting numbers, propagules of the pathogen in the soil. And then we wanted to submit a portion of that to Trace Genomics to get their assessment of soil health, looking at the whole community, all the pathogens in the soil, but particularly Fusarium Oxysprum lactucate. And then we wanted to compare these results and describe the soil population, the soil distribution pattern in the field. This is really important. This is one of the most important things we wanted to do in this study, is to describe the pathogen distribution pattern. Why is this important? If we've already mentioned that we can get a clustering of hotspots in the field where we've got a pathogen here but not the pathogen there. How are you going to sample your field if you've got pathogens in pockets in the field? How are you going to sample it in order to send in a sample and have someone give you an assessment on soil health, field health? Well, if you sample your field but you miss those hotspots and turn in that sample, they're going to say your field's clean. If you sample a field and you only sample those hotspots, you're going to turn in that sample and someone says your field is really bad. So you need to have an appropriate sampling strategy for your field in order to get an appropriate assessment of soil health. What is the population, including both the blanks and the hotspots? So you have to sample in a pattern in order to ensure that you pick those up. But what if the soil population, the distribution pattern in your field is uniform? It's the same all around. Well, that makes it easy. You just have to walk out to the field and take one sample and turn it in. So the pattern of the pathogen in your soil is very important in determining how you are going to sample that soil. And so this is one of the first steps to being able to assess field soil health. All right. Here's some of the information we've determined, and I know this chart, I'm not going to go through much of this chart, other than we've got our three fields right here. A research field and some other fields, both in the Yuma and the Welton area. And we have here our total Fusarium counts. Steve mentioned Fusarium in soil, and he talked about total Fusarium in soil, and versus Fusarium oxysprum in soil, versus Fusarium oxysprum pathogenic isolates in soil. And there's a professor at UC Davis, a retired professor, Ed Butler, and Steve knows Ed Butler very well. Frank knows him too. He used to say, the most common fungus worldwide in soil is Fusarium oxysprum. It's everywhere. Every bit of soil you dig up, you will get some isolate, some strain of Fusarium oxysprum in there. That's what Ed Butler is saying. And it's probably true because when we, and it's true, at least it's demonstrated here. This is the total Fusarium, actually it's the total Fusarium oxysprum counts, because we were using a medium that we could differentiate Fusarium oxysprum from Colmorum, from Solani, from these other Fusarium species. And this is our total Fusarium counts. And then using some recommendations, we were able to look at, assess our total Fusarium oxysprum lactucate counts. And they were pretty high in these fields, but this really didn't reflect what trace genomics was picking up. Because we can see here, trace genomics, the relative abundance of Fusarium oxysprum lactucate. We have high in here, high and medium here, and low and medium here. So our soil counts using these methods were not really representing what trace genomics was determining in these fields. Here's an example of the kind of data that we would get from trace genomics. And I thought this was very interesting in that they gave us some assessment of botrytis, all in one sample. Assessment of botrytis, lactucate, phoma, some bacteria, pythium, rhizoctonia. And here's Fusarium oxysprum lactucate here. And this was a number of different soils in a hierarchical pattern sampled. And we can see that, you know, it's pretty high throughout most of the samples here. Okay, and it's in a sense kind of uniform. So if I was going to go out and sample this field, maybe I only need to take 2 samples. I've kind of got the field covered. Save me some time, save me some money. Look at the difference here between this field and this is another field. This was our low population field, or our field with a history of low incidence of Fusarium wilt, and see what we find there. Only 2 of our samples, 2 of the 8 samples that we brought into trace genomics, did they find the pathogen in. And if we looked at some of our, using the trace genomic data and do some statistical analyses on these indices of clustering the variance to mean ratio, Loeys index of patchiness, Morositas index of dispersal. These are metrics that you can say how patchy that is in the soil. Is it uniform or is it patchy? And we can find that our research field, which had the highest incidence of Fusarium in the field, actually had the lowest incidence of patchiness. It was more uniform. So this shows that when our field first becomes infected, it's very patchy. And it's very hard to sample to get an assessment of that field. But as the field becomes more and more problematic, which means the population increases more and more, which means it's probably distributed more across the field, it becomes more even. And it's actually easier to sample a field that's really problematic than a field that's sort of in between, that might have a problem, but you're not quite sure. And those are the fields that you're most interested in. Because if your field is really bad, you kind of will stay away from it. You know it's bad. But if you're unsure, then you need to sample very clearly to be able to assess whether your numbers are going to be correct or not. Okay. So we're continuing this work. And so 2018, we have another grant in. It was approved, Arizona Department of Agriculture Specialty Block Grant Program, and it is ongoing right now. So I'm just going to let you know what our objectives are for this study. And again, we're going to continue our greenhouse work, because we think our greenhouse work is optimal in many respects, because we can control the parameters, we can control how the experiment is conducted, perhaps more tightly than if we conduct it in the field. One thing we want to do is, we want to improve the rapid detection for our LAMP and our RPA techniques, right? So we're continuing that with different soil types. We found that the soil type influenced whether we got disease expression or not based upon whether we infected it. So soil type is another factor that contributes to whether disease is expressed or not, whether you get symptom development. You may have an infected plant in one soil type with no symptoms, and you may have an infected plant in another soil type, and it's going to go down very quickly. So soil type seems to impact how that plant responds to the pathogen. Okay, we also wanted to improve our methods of inoculation in the greenhouse, because we think if we could dial in these techniques we use in the greenhouse for manipulating the pathogen, for causing disease reproducibly, consistently, we could use our greenhouse studies as a great way to screen cultivars perhaps, or screen products, chemical products, biological products. So we want to dial in our techniques used in the greenhouse. And then finally, this is where Rosa comes in, using if we can develop these methods for rapid detection using either RPA technology or LAMP technology, then hopefully we'll have develop some web-based tools that you guys can use, that the growers can use to implement these detection technologies in your own practices. So that is what we are doing at the University of Arizona over the last few years. And again, I will thank you all for attending, and I guess with everybody else, we'll answer questions when we're done.
My question is, what are the chances with this race 1 of seeing variability in pathogenicity? Is there any evidence of that with any other oxysporums?
The question about variability of pathogens, just race 1 alone, I mean, we have not looked at that. So I think all of us agree that in these pathogen systems, it's always possible. So it's not a very satisfactory answer, but I mean, especially for plant breeders and such, I mean, that's a factor you're going to have to look at. But we've done that with other pathogens, and there are variables there to factor in. So it's possible, but I just really don't know. Yeah.
Thanks, Steve.
Anybody else?
Well, that's one of the reasons we want to genotype isolates, so then we have some general idea of what it is that we're working with based upon a molecular level.
Any questions from the audience?
I got one. This is for Dr. Pryor here. So you suggested that some soil types can be pathogenic while others aren't, or soil type could be an indicator or a factor in pathogenicity.
They contribute to a plant's response to the pathogen.
Correct.
Yes.
So along those lines then, have you considered what other soil microflora or fauna could have either antagonistic or a competition effect on
Those are great questions. Those are great questions. The question was, so if we think that the soil type, whether it's going to be loam, whether it's going to be sandy, or clay, is going to impact a plant's response to a pathogen, and how about the other competing or symbiotic microflora in the soil? That's a great question. We haven't looked for that, but we have a grant in with Ryan Preston up at ASU to do whole genome or community community sequencing of the lettuce root rhizosphere. And for those kinds of studies, we will be looking not only at the pathogen, and we'll be inoculating these plants as well, not looking at the pathogen, but also looking at all the other pathogens, all the other microorganisms that are around the root, and see if we can get some sort of correlation between the presence of those other microorganisms and both the infection of the lettuce plant and symptom development. Because remember, symptom development and infection are two distinct processes. Of course, you need infection to get symptom development, but you can get infection without symptom development. So we need to look at both soil type and the microbial community to see that impact.
There's further complications that go with that. I think there was a grant proposal was submitted through here a couple of years ago, and one of the reviewers commented that there was some work done in Europe taking a look at certain groups of organisms that seemed to influence the level of disease that was subsequently found. I don't know how many other people have looked at that. If you go back in the literature about 30 plus years ago, there was a student of mine in Florida who had actually found that there was a strain of Fusarium oxysprum that can colonize watermelon. And it can systemically protect the plant from the form specialis niveum that can cause disease. So it was a non-pathogenic oxysprum, but it triggered the host defense reaction.
Sure.
So when the pathogen was present, nothing happened. I think there was some work in doing that at a Deb Fravel's lab up in Beltsville to try to commercialize that, because a student went there to do his postdoc. I think it kind of died somewhere along the line, and I never knew why. But there's certainly other organisms that can influence what goes on disease. The problem is, when you're trying to take a look at something like this in a natural system, what works on your research station here, it might not be the same 10 miles down the road, and it very much won't be the same thing in Salinas. And so that's what complicates taking a look at things like that. You know, people spent a lot of time in the biocontrol area saying, well, Trichoderma hartzianum is a great biocontrol agent because it worked in this spot, or this fluorescent pseudomonas worked best in this spot. But they're not finding that it works in other places because ecologically, they're not necessarily suited to be there. So it isn't saying that you shouldn't look at it, it's just, it's complicated.
That idea of cross protection, I think, is very important. The idea of cross protection is very important that Frank mentioned. A plant responds to both pathogens and non-pathogens as though it might be being attacked, and it produces all kinds of defense mechanisms to mitigate those interactions. And you can have a mild strain of a particular pathogen that may induce the plant to throw up these defense mechanisms and then further protect it from subsequent infections later on.
I have a question for Dr. Berry. So as context, we had the chance, when I was with UC, we had the chance to work with Frank on that RPA method, and it's a great method. It's a very fast, easy to do, DNA-based method for labs, and as a commercial lab, we use RPA a lot now, primarily for the strawberry pathogen. So did I understand, can you clarify, you are trying to develop an RPA or LAMP assay for the lettuce Fusarium? And
both RPA, yeah, both using the AgDia platform and the New England Biolabs platform.
So I don't understand, does AgDia have one for lettuce already?
No, we're developing the primers and the probes for that.
Okay, right now it's not ready for prime time, it's not working well?
It works. I guess we could just, it works, but like I showed, we had false positives. We did not detect every infection that we had. So it's not ready for prime time.
Okay, well, just to follow up on that, what we have found in the strawberry system, several things. One is where you take the tissue to sample is extremely important, right? We had students, the postdoc who was working with me, they went through meticulously and looked at different parts of the plant and where they did the recovery, where they got positive results, where they didn't. So that where you're taking that sample is very, very important. The stuff with the RPA, it's not like the real-time PCR where there are very firm guidelines on how annealing temperature affects specificity. We've worked a lot with the people at TwistDX in the UK, and actually the postdoc in my lab, Tim Miles, that developed this for Phytophthora, ended up visiting them and was in their lab for a couple of days, getting a lot of background information on exactly how this all works. And basically it's a black box. They don't even know what you have to do to make it specific. So we've gone through in a lot of our work and trying to collect data when it doesn't work, so then we can model it to try to come up with something that says, this is what you need to do. But in comparison, we had markers for 2 highly regulated Phytophthora species, Phytophthora morum, which causes a sudden oak death that is all over California and in the nursery system, and then Phytophthora kernovii that is not in North America, but is in the UK and other places, and the regulatory people don't want it here. So when Tim Miles designed those markers, the one for kernovii, the first time, boom, was perfect. The one for Ramorum, he had 1 forward primer and tried 10 reverse primers before he got the specificity. So I just encourage you to try a range of different primers. The primers are cheap. It's the probes that'll kill you. The probe alone is over $600. It's very, very expensive. But the primers are cheap. So go ahead and try a range of primers and try different things with it to figure out which one works. Because there isn't an exact science where you say, well, if you have a base at this end of your primer, if it's different, it won't work. In real-time PCR, that's correct. In RPA, it is not correct.
There are programs for primer designs that work very well for conventional PCR, but when you're working with an isothermal platform, it can be much more challenging.
It seems certain rotation crops can suppress Fusarium, but some Fusarium continues to grow right in the field, right alongside the new crop. It's not showing symptoms, but it's still growing in the field. What are the best crops for rotation?
That's a Steve question.
So that's a great question. So we've done a lot of work with that in Salinas. So that's the footnote, is this is done in Salinas, but I think there's promises here. So the thing that's really funny is that broccoli thing. So we talked about the broccoli roots, and we're really upset to see that. However, doesn't mean you shouldn't use broccoli, because maybe the way broccoli operates, you would overcome that. And meaning, if you see a lot of our research, we're really high on broccoli. I'm high in a good sense, Mike.
Okay.
We're very in favor of you doing broccoli, because broccoli, when you grind it up after you plow it in and everything, it really suppresses soil pathogens significantly more than cauliflower, cabbage, some of these other things. So broccoli has those chemicals in it, those glucosinolates at a high level. It really suppresses pathogens. It's a great mechanism. A lot of organic growers in coastal California use broccoli because of that. So I think that would probably counterbalance and overcome that problem with the root colonization I talked about today. But we've not actually tested that clinically to prove that. But I would say that's an example of looking at previous research and saying, you know, that is a crop rotation option that's probably going to be very helpful based on previous work. And then someone's got to, you know, someone's got to do that down here and verify that. But that's one. And then the other things too, you know, the grains still are very useful. They have, like Sudan, it's not a super suppressive. Broccoli's better. Sudan also can suppress things. But those are things, again, based on previous research, we would say those are good bets to try. And then you've got to take it from there. Your research has got to take it from there. Actually, a question for Stephanie or Paul. I mean, so we share with you our California concern, and it's a rising importance for us. I'm just curious, are there already linkages with the California Lettuce Board, or you talked about things in terms of mutual progress, mutual projects, or working together collaboratively, anything in that area?
I haven't been in contact with them, but I am going to their conference, the Calgary conference in March. So I'm going to try to form a relationship with them. And I know Paul has.
Yeah, we were linked somewhat with Mary, but and then when she left, we kind of lost that linkage. And so Stephanie's going to build that back up again with the conference, with her replacement.
And I've spoken to lots of California researchers who have grants through them, you know, Frank and you and Tom Gordon and Richard Mitchell Moore. And so
there's probably a lot of room for collaboration.
I think so.
Yeah, there is a lot of room for collaboration, and hopefully we can get some projects going with them.
Quick question for Barry, if that's okay. Yeah, you were talking about Fusarium in the flower heads, and you said that it doesn't reach the flower heads. How high up in the plant
It
does
does,
actually?
it does reach there, but at poor recovery rates. In other words, we can follow it right up the inflorescence, and down in the crown.
How are you following it up the inflorescence?
Sampling the parts of the plant and then plating on semi-selected media.
Okay.
And up at the inflorescence, most of the times we plated, we would not recover it. And this is disinfesting the surface of the plant, so you're not getting anything that might be adhering to the outside of the plant. This is stuff that's inside the plant. And of course, we never recovered it from seed. From embryonic seed tissue, we never recovered it from there.
Okay.
But all you had to do is mix clean seed with a little bit of trash from an infected plant, and the whole seed lot's infected.
So if you hand harvested the seed, you wouldn't be able to find it, but if you thrashed it,
Oh, that's a good point. So, but that would be, wow. So yeah, I'm sure in experimental lots, yes, you probably have to go in and clip,
Oh,
make sure
this-
nothing on the bottom gets touched.
that's my question, because we're a breeding company, and I'm trying to figure out what's the best way to harvest seed from, obviously, plants that might have been grown from a Fusarium, or
Correct.
selected from Fusarium field, so
Yeah.
without transferring the Fusarium.
And Steve has worked with vegetable seed as well a lot. Much of the pathogens on vegetable seed are on the outside. The seed is infested rather than infected. And usually this comes about through the threshing process.
So you're thinking that hand harvested seed would be okay, whereas threshed seed would?
If the plant's not infected, threshing's fine, but you've got to make sure the plant's not infected.
It's a safeguard right now to do it that way.
Right, it's just, yeah.
But I'm asking if the plant's infected.
You'd have to look at each and every plant, because you can't look at.
Test it.
Yeah, test it, you couldn't look at, as Steve showed, you can't look at individual symptoms, because they're gonna be, there's gonna be symptoms.
Fish seed plants, right? They look like hell at the end. So.
Yeah, right.
Could a seed lot that could potentially be exposed that way be cleaned up via fumigation or anything?
Those seed treatment protocol are well established, whether it's hot water, bleach, or even a fungicide, some sort of fungicide protectant.
Yeah. So probably yes, right? You could probably clean up the external part of the seed.
But then you'd want to test afterwards, of course.
Yeah, have you guys done any comparisons between Fusarium that you guys find in conventional fields versus organic? Or is it pretty even across the board?
Yeah, we've not. It seems the same problem, same issue. I don't know who has it worse. I've not looked at that. But in terms of the disease, the severity, it seems to be no difference.
Okay, I have a question for Steve. So, after oil have different races, right? So you have race 1, race 2, race 4. And have you seen any differences in terms of symptom expression? For example, certain races only cause root rot type, and certain races only cause maybe crown rot,
Yeah. So based on this question, we have not, because again, in the West Coast, includes you, so far we only believe there's only race 1. So because of that, no, I have not looked at differences between the races because we don't have the other races. And the most we could do, we could all do this together, is to look at reports from other researchers and see if they've done that comparative research, or if they describe the symptom profile for race 4, and is that significantly different from race 1. So based on reports, we can compare that. But no, we haven't, because fortunately we've not had the chance to compare them, in hand. And we hope never to have that, right? Right, everybody?
Yeah.
I have a question for you. So, can you give us an idea about the cost of the RTRPA, the RPA field system, apart from the $10,000 that we spent on that small box? What are the other costs?
More about the actual, you would know actually more about the better. I know what we charge, right?
We've only worked on it 1 year. And it was expensive.
So I think
Make some money, man.
where? I don't know what detector you get. You can use a real-time PCR machine to detect it if you're doing a fluorescence detection. You can get Agdia cells, their own meter to measure the fluorescence emissions. And it sounds like you've already purchased one for about $10,000. The
I
kits.
just know how much it's cost. I want something that I can take to the field. So I wonder.
Well, there's another company, they used to call it the Bio Ranger, and they changed it. I guess it's now called the Bio Ranger. It's a company in Hawaii, and it runs about 3 grams. And it's about 3 times the size of a pack of cigarettes. It's initially was designed for loop-mediated amplification lamp assays, but you can program it to work for RPA assays. It has 2 channels it runs off of, and it is fully portable in the field. Again, you use an app on your smartphone to run the thing. It's pretty slick. For the cost of using the reagents and stuff, the RPA kit is an interesting thing. It comes in 8-strip tubes, and in the pellet, everything that you need is localized in the bottom. The difference between the stuff in Agdia and TwistEx holds a patent on it. And Agdia has licensed that patent, and they're doing their own formulation. So they do their reactions in 25 microliters, TwistEx does it in 50. Each of them says theirs is better. We find they're pretty much comparable. Each one of those tubes runs about $5. So it's not cheap, and it's going to be about $1,000 for the primers and probe. But you can pencil it out. I don't know how many thousands of amplifications you can do with that. I mean, Steve would know we haven't done that. We've done it from a research standpoint. So it's not a low point in entry, but I imagine your total costs, materials costs for doing this, we use a regular ELISA buffer from Agdia that's not that expensive. I think our total materials cost per tube per assay probably runs somewhere around $5 to $8, but then you also have to include controls and do other things, and sometimes you can multiplex them. Agdia does have a portable unit with a battery that you can take it out into the field and use, and that's about $7,000, I think. And they have something that is basically set up for more commercial things like Steve does. They have a barcode scanner that you can go ahead and program your protocol and make a barcode. So if you're changing between different diagnostic assays, you don't have to reprogram the machine. You just take that barcode, put it in front of there and hit it, and it automatically changes that machine to do that package.
And of course, as we've heard, the problem with the RPA is not ready yet. We're waiting for Barry to set up the RPA primer so that it's reliable, right? No pressure, but maybe tomorrow? This year.
I have a question for Mike, I guess, since he's the veteran Fusarium Wilt researcher. Is there anything as far as cultural methods that you've seen that you're interested in, excited about? Anything you can say about methods to reduce the population in the soil?
Actually, Matt is in the audience. He's done some work on different kinds of cultural methods, and to make a long story kind of short, nothing really worked that well. But why don't you give it to Matt? He can tell you. Last year, actually, they did different kinds of cultural studies, very extensive. And I, just off the top of my head, I kind of remember the results. But would you mind commenting on that?
No, we tried fallowing the ground. We tried deep plow, 32 inches. We tried medium plow, 22 inches. We tried a regular 14-inch plow. We tried a bed cooker that on the preformed beds with moisture and without moisture, and cooked the beds. They, off the back of the machine, they came out about 240 to 260 degrees. Make a long story short, none of those worked. Fusarium came right back in those areas. I will comment, somebody asked about cover crops. Steve Sedan looks like it lights that Fusarium on fire and makes it even worse than what we started with. So I would just be a little careful of Sedan right off the top. We have some anecdotal evidence that it is just like Fusarium on crack or something. It goes crazy.
So you guys done ASD and all, you mentioned that. ASD, is that something you guys are considering?
I mentioned it because I'm trying to put a project together, but you can talk more about that.
Actually, that,
Oh, that's it.
I got
well,
if I could just make one comment on that, there has been some work on ASD on strawberries, and one of the concerns is it really dramatically increases the Forma specialis frigariae because it colonizes the organic substrates that you're adding in. The Fusarium oxysperms are very aggressive saprophyte. So at least in the strawberry system, they're finding
that it's increasing the populations, which is of concern.
In Salinas though, when
Yeah,
you're
in lots of
not really able to cook it.
Right.
Yeah.
I just had a quick one. So basically, if you've already tried fallowing, so you'd have to fallow a number of years before you would reduce those numbers. And there's no other vegetable that we grow down here that is not a host for that you know of. I mean, it's still it's surviving through it, and if it's actually increasing, the amount of Fusarium out there, I'm just wondering, there's nothing we know of that you can rotate that field out of with another produce crop that we grow down there.
Bill, to say fallow, we ended a lettuce crop in November. We did absolutely nothing to the field the following November, and the field lit up pretty good.
So that was one year.
Almost one complete.
Well, Tom's Gordon's data suggests maybe 3, 2.5, 3.
Well, that's fine.
So that's a little hard to do.
Yeah, that's kind of hard to do, right.
Isn't that a study you referred to?
Hay alfalfa seems to reduce it some.
Okay.
We've had success with very, very bad fields, and after 4 years of alfalfa, grown a lettuce crop. And we're trying not to go lettuce, so it's been lettuce to broccoli to other things, back to lettuce. We won't go back to lettuce for another year. So, but at least the first crop would let us right back on it. It turned out perfectly fine.
We just need to figure out a way to keep the alfalfa market up for 4 years.
So maybe the answers are in, as we've been talking about in other forums, soil health management, either promoting or developing certain soil types or farming in certain soil types, or developing the soil microbial community to such an extent excuse me, that it suppresses or induces the host defense systems or something along that line.
We're playing around with, I haven't talked to Paul yet, but we are going to be playing around here shortly with different cover crops. Not cash crops like sedan or anything like that, but we have a number of crops we can grow through the summer. And then just come in and see if any of that has any effect whatsoever.
Sedan's a cash crop?
Well, it used to be a cash crop. It's kind of break-even crop.
And the thing about cover crops, some of our Salinas Valley growers are merging the concepts or categories. So some of them are planting broccoli as a cover crop. They find cheap open pollinated varieties and plant it densely like a cover crop, and that accentuates that suppressive action I talked about. And I feel that is going to counterbalance this root colonization thing that we found. I think that the benefits from broccoli residue will overcome the little bit of colonization on the root. That's my feeling. And maybe you guys can consider that as a research thing. But the biomass is important on broccoli. So grow densely, grow tall if you can, and that's where we get the best results in the coast anyway.
Was there something with how you prepare the broccoli? You don't just disc it under, do you?
Yeah, just disc it under.
You're not mowing it up to chop it up into smaller pieces?
As researchers, we did that, but we're trying to, we did extension research. We're trying to get practice for the growers, so they'll flail mow it or they'll try and break it up. So yeah, they try their best to do that. We ground it up, really fine because we could do that as research. But,
So the number of crops that have been reviewed as potential suppressive crops, 10, 12?
in terms of
the number of crops, because you've mentioned broccoli, cauliflower, sedan.
Gotcha. So suppressive crops, at least based on our work, we're going to take Sudan off the table, but Sudan, of course, for other systems. Been tested, yeah. Sudan's been tested enough for Fusarium, for
I'm
herbicide.
just thinking there must be a slew of other crops that could be tested.
Test a bunch, but if you want to cut short the chase, look at the brassica group. Okay, so we know that broccoli is the best. Brussels sprouts, interesting, has suppressive effects. Cauliflower does not. And if you look at the chemical profile of these things, botanically, they're so close together. Broccoli and cauliflower are worlds apart on the glucosinolate. So broccoli, hands down, number one. Cauliflower, don't even consider it. And then Brussels sprouts. No, because we're talking about suppression.
But in terms of colonization.
Oh, colonization was just those three, Barry. Broccoli, cauliflower, and spinach.
Some things to think about, because if we're talking about going fallow, we can dry fallow, and that's cost prohibitive. We're not going to do that. But if there's other opportunities to fallow and something else, maybe those crops could be
But it also could tell you what not to rotate with. If something brings on a higher percentage, it could tell you what not to rotate previous to lettuce.
Yeah.
So, I mean, that could be a factor telling you what not to use beforehand if you plant lettuce, or what not to.
Those seem like good areas of research then.
Yeah, but the suppressive thing, look at the brassica group. And the last thing I was going to add to that is mustards. Mustards as cover crops, they do have a suppressive effect, but it's way inferior to broccoli. And in fact, some of those, those gold variety, whatever they call those, we're a little lukewarm on those compared to broccoli. But if you look at your ads, they're really pushing mustard cover crops for suppressing diseases. There is an effect, not as much as broccoli.
So Steve, do we know what the mechanism is in broccoli that's suppressing it?
It's pretty well understood. There's two parts. It's two parts to that, and then Christian and I did some of this work, but there's a lot of other people, so it's not our work. There's two parts. One is it's chemical. So those glucosinolates, it's actually an analog of FAPAM. So that is suppressing it, and then it definitely enhances the microbial population and the mix in the soil. That's suppressing these pathogens. So those two things out of the broccoli and crucifer group, that's definitely what's happening. So it's a very interesting phenomenon.
So is there any incentive to look at different varieties of broccoli compared to each other?
Definitely if you have the time and inclination. I mean, yes, that's really a fine point. I would say it's broccoli versus the other things. If you guys need to do cauliflower, well, we already know cauliflower doesn't do it, so don't bother. So yeah, I'm sure there are differences in the varieties. We didn't have the luxury of time to do that, and our Slinski Valley growers, same thing. I mean, it's time and money out the door, so we just go with broccoli. And we were working more on how to use the broccoli. Again, high biomass, so grow it tall, and or plant density. That you'll get the best bang for that if you do those types of approaches to biomass. We had Man Packing, those guys. They took our idea and they grew broccoli as a cover crop. They grew it to about 6 inches tall and plotted under. There's no effect. Okay. So again, it's plant density and the amount of biomass. So grow it big or grow it densely.
Did anybody take it to flower?
I don't think anyone did, and that's an interesting thing. If that changes, if it improves the return on that. We wanted to make this, we're extension people, so we were trying to make it practical. So we grew it to harvestable state size, because our pitch was grow broccoli, and if you could sell it, harvest the heads, and then plow it under, and that worked great. If you can grow it taller, fine. We never went to flower though, I don't know about that. But we wanted to say, let's make this practical. We grow 35,000 acres of broccoli in Monterey County alone. So it made a good fit if you could sell it as well as plow it under to suppress verticillium. And therefore now, by extension, can we suppress Fusarium with that? That's the idea here.
Does broccoli also work, I'm sorry. Does broccoli also work better than mustard on Sclerotinia?
Yes, it does. We published some work on that. So our work has been on verticillium and Sclerotinia. We're very high on using broccoli for that. Mustard in the same study was mediocre. It's okay, but if you have the choice, do the broccoli. And again, I want to emphasize, we're proposing that you or someone look at that for this Fusarium, but we did not do Fusarium, right? At that time, it was just a desert problem, remember?
Question. I see you guys are doing a lot of work on markers to identify it in the field. Is there any work done to identify the gene or genes within the varieties for breeding purposes?
I know that it is for strawberry. Whether it's for lettuce, I don't know. That's a Richard Mitchell question. My suspicion is they probably are looking at that. I wouldn't be a bit surprised if some of the breeders at our Salinas location, they've got quite a germplasm collection. I wouldn't be surprised if someone's taking a look at that right now. I can't say it for a fact. If you want to shoot me an email, I'll see if I can identify with some of the breeders if anybody's looking at that. And just to add one quick thing into what Steve was saying, it was my understanding that glucosinolates accumulate more in the plant the older it gets, which is one of the reasons why you want to let it get big. And it might not have worked when it was done small because you just didn't have the biomass. And I spoke to a TNA grower, and he tells me that it works great for managing their problem in cauliflower, the verticillium of cauliflower. And they routinely, when I spoke to them, they would come in with a certain percentage of their land every year and just drill in like Merced Ride, the open pollinated broccoli seed, and put no inputs into it, and just harvest it for biomass and put it in the ground. And he said he did a really good job of managing the verticillium wilt in cauliflower.
Frank, you know, and I like what you say because, and Steve, one of the things that we would like to do is to be able to use a crop that we can harvest. And this is my question about the varieties. Because if we know what the mechanism is, yeah, maybe it doesn't need to be taken to flower, but that question needs to be answered.
Some broad varieties. I have a question for you guys and maybe you local expert, but are you finding lettuce varieties that are tolerant or resistant that are commercially available, so you have some options for that? Because I forgot the name, but there's one romaine that we use in South Salinas that has some tolerance, so they can get by with it. It gets diseased, but it's tolerant. You guys have choices like that commercially for the short term? You can plant some things that hold up a little bit?
Certainly. Better ask the grower.
Yeah.
Well, you know, as far as romaine, we had some in our trials this year, and you know, they weren't immune, but it was a nice harvestable stand for varieties of romaine. One was Del Sol, the other two, one was, didn't have a name yet, the third one, name escapes me right now. But it basically, those look very nice, especially next to the crisp head lettuce next to it that did not look well at all.
Any crisp head available?
There is a couple. You could ask the growers if, you know, if they would fit into their growing plans, Oracle and Meridian. And another one that looked promising this year, it doesn't have a name yet, but you know, it's only 1 year's data on that. But the Meridian and Oracle definitely are ahead above all the other varieties. Now, whether they fit into the scheme here or not, does any grower, have you tried these, and what do you think?
Little small, not the best. We do have a couple other commercial varieties that we went through screening, looked very, very good. In the plots for some of the different plant breeders, they have some stuff coming up that looks very good. Most of the romaines showed almost 100% resistance. Not all of them, there's a few that
Yeah, there are a few that, yeah,
and
look
most
like
of the
dogs,
desert
yeah.
varieties are resistant.
And these are in fields that with the history, right?
Heavily.
Oh, okay,
Yeah.
very good.
Yeah, this is in our
So they were
research.
challenged.
They were.
Thank you very much.
Steve said that's it. That's it.